Hi everyone, this is your instructor Kwame Yushen, and this lecture is about the introduction to taxonomy and classifying microorganisms. In terms of the objectives, we will look at taxonomy and phylogeny, the characteristics of the main domains, bacteria, archaea, and eukarya, the differences between eukaryotic, prokaryotic, and viral organisms, scientific naming and the importance of it, and why it stands out compared to um, normal naming as we find in different languages. Differentiating between a culture, a clone, and a strain. Be able to tell the difference between what classification and identification is. The importance of identifying microorganisms. We'll also look at Berge's manual and the two types in terms of the main types of Berge's manual and what the emphasis is in terms of each of them, their purpose. Staining by chemical tests and the application in identifying microorganisms. And we'll also look at serological tests, phage typing, flow, cytometry, and as well as some DNA techniques that are used in classifying and identifying microorganisms. And how we can apply all this in making a dichomous key or a cladogram. So for taxonomy is the science of classifying microorganisms based on their characteristics, and until more recently we were just looking at broadly or grouping organisms based on plants or animals. So the groups that we form in taxonomy, in terms of this science, the groups are called a taxa. So a kingdom is a taxa, a phylum is a taxa. So phylogeny or systematics is a taxonomy that uses evolutionary history of organisms to group them. So when we use evolutionary history, we are applying phylogeny or systematics. And in terms of the hierarchy, if we use phylogenetic relationships, which is what we use now, then we will have different hierarchies. We'll look at that briefly. But historically, in 1735, Carl Linnaeus devised a two-way system of classifying organisms into plants and animals, and we very soon realized that those classifications are not inclusive enough. It's too vague. And so in 1969, organisms were grouped into 5 kingdoms. So you had the kingdom bacteria, you had fungi, you have plantae, you have protists, and animalia. And then that system was also not all encompassing once we discovered archaea bacteria. So the discovery of archaea bacteria led to a new system, the 3 main domain system that we currently used. But it's more because we were able to tell that there were differences between the RNA sequencing of archaea bacteria and bacteria. So we could tell that there were 2 prokaryotic cells that existed. More recently, in 2000 to around 2000, we had a project called All Species Inventory Project that was started by Kevin Kelly and Stewart Brand in San Francisco, and the goal is to identify and name every living thing all in a single human generation. It's a very tall order. It started a foundation in 2001, to do this, relying mainly on amateur scientists and scientists all over the world volunteering their time to do that. The three-domain system has bacteria, archaea, and eukarya. And it is based on the concept, the evolutionary history, that living organisms started appearing about 3.5 billion years ago, and that they all have a common ancestor. So the first cell that appeared or originated eventually evolved into the different groups of cells that we have. So we have bacteria with its own unique characteristics, archaea and eukarya. And this is an example of a cladogram, and it's based on the RNA sequencing. RNA, ribosomal RNA sequences are highly conserved, and so even if mutations take place, the mutations are significantly very low. So we can actually tell how connected these organisms are. So based on the three-domain system, these are the basic characteristics that we're looking at. There are more, but these are a few of them. And based on the cell type, you have prokaryotes, so those that do not have a membrane-bound nucleus. So you have archaea, and you have E. coli or bacteria, all being prokaryote. And then you have those that have membrane-bound organelles. Those are eukaryotic based on the cell type. For cell wall, there is variation in archaea, but they have no peptidoglycan. For bacteria, have peptidoglycan. Eukarya will vary based on whether they are fungi or plants, and then the cell wall composition will be different based on that. In terms of membrane lipids, archaea have branched carbohydrate chains that are attached to glycerol. Bacteria have straight carbohydrate chains or carbon chains that are attached to glycerol. And eukarya also have straight carbon chains that are attached to glycerol. And then for the first amino acid that are synthesized during protein synthesis, archaea have methionine, eukarya also have methionine, but bacteria have formyl methionine, a variation of methionine. Generally, we will say bacteria or bacteria, they are the virtuosos. They are very highly skilled in using nutrients and harnessing inorganic molecules to synthesize food, mainly for the sustenance of food chains. Archaea are the weirdos. They live in very extreme conditions. And the eukarya are the predators and the thieves. And this, I say with a pinch of salt, in that there are exceptions, because if you look at plants and some protists, they're able to synthesize their own food, so they don't have to depend on other organisms for their food. So that brings us to the endosymbiotic theory, the origin of eukaryotes. That a current example is a Cyanophora paradoxa, which is made up of a protist cell. So there's a protist cell with a bacterium that lives within it symbiotically, and it gives more credence to the endosymbiotic theory, that at a point in time you had bacteria, aerobic bacteria and cyanobacteria, that were engulfed by a regular cell, which just was like a bacteria, formed a membrane around its nucleus over time, and eventually engulfed anaerobic bacteria, which became mitochondrion, and then a cyanobacteria, which became chloroplasts. In terms of the scientific name, the advantages that it has over common names is that there is no variation, so it's used worldwide, and it doesn't vary in terms of geography as well. If we look at how different cultures name different organisms, it's based on the language, based on their understanding. And so without more or less a universal language, as in binomial nomenclature, there will be a lot of confusion when we're trying to communicate with each other in a global setting as we do now. How the organisms are named based on the genus and species name, as in how the genus name is sourced and what the species name is based on, doesn't really follow any strict rules. For instance, you have Klebsiella pneumoniae, where the genus name honors Edwin Klebs, and the species name describes the disease that it causes, which is pneumonia. If you look at another example, Streptococcus pyogenes, where the genus name is sourced from the shapes, the morphology of the cells. They are straight chains, and so strepto, and then it forms a pus, so that's a pyo, so Streptococcus pyogenes. So there are no hard and fast rules about it, but the bottom line is that there is a genus and a species name that are accepted by internationally recognized bodies, and then it becomes a universal name for that particular organism. So let's look at a few definitions here. So eukaryotic species are a group of closely related organisms that are able to breed among themselves. They're able to reproduce, and their offsprings are fertile. For prokaryotic species, we are referring to a population of cells that have similar characteristics based on Berge's manner of systematic bacteriology, which relies on the ribosomal RNA sequences. And so under prokaryotic species, we have culture, that is organisms that are grown on a media in the laboratory. You have clones, a population of cells that originate from a single cell, so the copies of the same cell, and those are clones. Then you have strains. So these are cells within a clone that genetically are different from each other. Then we have viral species. Viral species refers to population of viruses that have similar characteristics and are found in a similar ecological niche. We don't place viruses in either the domain or the kingdoms because they are not cells and they cannot grow without a host cell. So if we look at the relationships in prokaryotes in terms of the phylogeny, in terms of the evolutionary history, we see that there's a common ancestor, and that this common ancestor, which appeared about 3.5 billion years ago, has evolved into groups of organisms that we will describe as bacteria based on the cell structure and the ribosomal RNA sequences, and those that will group under archaea. The two are more related than eukarya is related to either of them. You will notice that we can use the content of glycogen, guanine, and cytosine in terms of the nucleotide base pairs to actually group bacteria, gram-positive bacteria. Pretty cool. Then there's the domain eukarya. In the domain eukarya, we have animalia, which are multicellular. They don't have any cell walls. They are chemoheterotrophic, so they rely on other organisms and a source of their energy and a source of the organic carbon molecules as well. Then we have plantae. They are also multicellular, but they do have cell walls made up of cellulose, and they're usually photoautotrophic. They can synthesize their own. We have fungi that are chemoheterotrophic, unicellular or multicellular. The unicellular ones are yeast, multicellular are molds. They have cell walls made up of chitin, but they develop spores and hyphal fragments as well. Then we have protista, and that's eukaryotic organisms that do not fit into other kingdoms, so they all clump together. So you have the The amoebas, the euglenas, the parameciums, and all that, they all fall into this category. So they are grouped into clades based on their rRNA sequencing, the ribosomal RNA sequencing, which are highly conserved. So what's the difference between classification and identification? So classification, what you're doing is that you're placing organisms into groups based on their relation to each other, their evolutionary relationship to each other. So we list the characteristics of the organisms that we know, and then we're able to compare the RNA sequences and tell which one is more closely related to the other. Then there's identification, where you match the characteristics of an unknown organism to a list of known organisms, with the goal that the unknown organism is an organism that we already know, and that based on the results that we get from the test, we can actually identify which organism it is that we are looking at. Okay. So why is it important to identify microorganisms? Apart from just knowing what kind of organism it is, just for knowledge, it helps us in identifying food poisoning outbreaks, so that if there is an E. coli strain that is causing a particular food poisoning, we can actually trace quickly the source of the E. coli, because we can identify them up to the serological level, the different strains that exist. You can tell whether it's E. coli 026 or E. coli 0157, and then be able to know how to deal with it. We can also use that information to tell if there is an antibiotic resistant bacteria. We can determine that to avoid food spoilage, botulism, and environmental exposure to microorganisms that may be harmful to us. And an example would be Naegleria fowleri. Naegleria fowleri is a protist. So it's an amoeba-like organism that is able to eat our brain cells. So if somehow it gets into your system and it's able to access your brains, it would end up getting infected, and it will result in fever, headache, nausea, and generally people die from it. So of the over 140 cases that we have known, only 4 people have so far survived this particular protist. So if you know that an environment, you're able to identify this microorganism in an environment, then those that live in that area can be warned about it, and then you will know the risks that are involved if you decide to swim in such a water. So if you assume that you isolated a gram-negative bacteria, it produces acid. When it ferments glucose, produces acid, and then it produces also a gas. And then it's also motile. So that's a motile bacteria at 37 degrees Celsius. What bacterium is it? And the way to do that is to form a dichomous key. So you look at gram reaction, is it positive or negative? In this case, it's negative, so that's a gram-negative bacteria. You look at glucose fermentation. Glucose fermentation, we have those that produce just acid, and those that are motile produce acid and gas. So acid and gas, so it goes here. And then in addition to that, if we look at motility, there are those that are not motile and those that are motile. And so for the example that we have there, this would be our bacteria. Aeromonas hydrophila would be the bacteria. So this is a dichomous key, and we can use that dichomous key to identify an unknown bacteria based on the tests that we run on it and the results that we get from those tests. Berger's manual is an important reference when it comes to identifying bacteria. There are 2 types of them. We have the Berger's manual of determinative bacteriology, and then we have Berger's manual of systematic bacteriology. Now, the one that is determinative bacteriology, it just provides information about identification. So it's more about identification using different tests, morphology, staining, and biochemical tests. And then the systematic bacteriology uses phylogenetic information. So we are looking at ribosomal RNA sequencing, and so it's more about classification than it is about identification. Okay. So those are the differences. There are different methods that we can use to classify or identify bacteria. If we can use morphology for identification, differential staining, biochemical tests, serology, phage typing, fatty acid profiles of the bacteria, flow cytometry, DNA-based composition can be used for classification but not for identification. And DNA fingerprinting can be used for both, PCR for both, nucleic acid hybridization techniques for both, and rRNA sequencing for classification but not identification. We'll look at a few of these. So if we look at morphology, you could look at just the shapes of the organisms, and in eukaryotes it's very easy to do that. But with bacteria, we have to go a step further. In addition to just the shapes, we could also look at how it stains based on the cell wall composition. So you have gram staining, acid-fast staining, and then you have biochemical tests. Biochemical tests, the underlying principle is to test for the presence of bacterial enzymes. So you provide certain substrate to the bacteria, and then you see the metabolic process in terms of the products that are produced. And since reactions are governed by enzymes, it then gives an indication whether the bacteria produces a particular enzyme or not. There is a rapid numerical identification test that is now available and done, where you have a number of tests that have all been put together in a tube. And then what happens is that this tube have all these different tests. You inoculate your organism into the tube. So you inoculate the organism into the tube aseptically, and then you incubate it. And then you would then come and look at the results based on the different changes that you see. The results are all grouped. Each group will have a value. So for glucose fermentation, you have 2. Glucose and gas, you have 1. So this for this organism is 2. For hydrogen sulfide production, it's 1. It produces it. For lactose test, arabinose test, sorbitol test, Voxter-Proska test or VP test. For all these, it's 0. And then when it comes to phenylalanine, urease, and citrates, it has all these values as well. So what it does is that you add the values up. So this is 0, this is 0, this is 1, this is 2, and this is 7. And so there is a computerized listing that you compare the figures that you get against. So in this case, you have 21007. And so 21007 is Salmonella chloriosus. So we can then use this to identify organisms fairly quickly in the lab. Otherwise, you would have to run each of these tests individually. Then you have serology. So in terms of serology, microorganisms generally would have certain antigens, proteins, and carbohydrates on the surface that would cause the immune system to produce antibodies. So we are looking at antigen-antibodies combination. So a combination of anti-serum and the antigens on the bacteria. Now, when you have such a situation where a particular antibody is specific for antigens of a specific bacteria, you have what we call agglutination. So there is clumping that forms when you mix them together. And it's this principle that is used in slide agglutination. There's an example of it. ELISA and Western blots as well. You will do ELISA in the lab, and so you have more details on that. And in terms of Western blot, the principle is that proteins are separated by electrophoresis and then can be detected by their reactions with the antibodies. So bacterial or microbial proteins are separated by electrophoresis, and then they will react with antibodies. And then if there is a reaction, you can tell that it's that particular organism that you are working with. Then there is phage typing. With phage typing, you would plate an organism. So you inoculate an organism on a plate, an entire surface of it, and then you introduce bacteriophages. Bacteriophages are viruses that infect bacteria. And so when you introduce the bacteriophages and you incubate it, and you end up with these results, what it means is that this particular strain of bacteria that you plated is susceptible to the phage. Now, phage are specific, are mostly species specific. So based on that information, we can actually group bacterial strains, the different strains within a particular species based on their susceptibility to phage. And you see all these clearings because there is lysine that has taken place. So the virus will take over the bacteria protein synthesis and go ahead and do create its own copies, several copies. They get to a point where the bacteria will then lyse and these species will be created. Then there is flow cytometry. Flow cytometry would use a moving fluid containing bacteria through a forced opening. So you have bacterial strains that have been stained. So you have the cells that have been stained. You have a sheet fluid. A sheet fluid is basically a saline solution. And so the sheet fluid is just run through, and both of them are run through this nozzle. And so they end up going in a single file. And so you have a light source that is directed towards it. And then you have a fluorescent detector. And so bacteria, as it passes through, will be picked up by the fluorescent. They will fluoresce once they pass through the detector. And then that information will then be used to describe the bacteria in terms of the shape and the likes. It is proposed as a quick method to detect listeria in milk. So if you run through it, we know the shape or morphology of listeria, and so when it's identified by the fluorescent detector, we can tell whether a particular milk has listeria in it or not. Then there's the genetics aspect of it. You have DNA fingerprinting, where the number and sizes of the DNA fragments that are produced by restriction enzymes are used to determine the similarities or differences. So it's run on electrophoresis gel, and once you run it on a gel, you can tell based on the sizes and the positions of the fragments whether two samples are the same, they are the same DNA composition or not. If we look at our example here, this and this looks quite similar. And so you can tell that they are closely related or it's the same organism that you're looking at. Then there is ribotyping, which refers to ribosomal RNA sequencing. So ribosomal RNA sequencing is very important in classification because it's a very conserved region of the nucleic acids. Then we have PCR, polymerase chain reaction, which can be used to amplify small amounts of microbial DNA in a sample. And then we can also use that to identify an organism. Once we're able to amplify it, we can then identify which kind of organism it is. Nucleic acid hybridization also uses the property of complementarity of DNAs to actually tell whether an organism is closely related to another, or it's actually the same organism they're referring to. And it can be used for both DNA and RNA organisms. So for instance, if you have a DNA organism A and then DNA organism B, so this is A and that is B, what we will do is that we'll apply heat to it and cause the two to denature. So there is separation of the bonds, the hydrogen bonds between the nucleotides for both of them. And then we'll mix them together. When we mix them together and allow them to form bonds, what we expect to see are one of these three. We'll determine one, if the two were able to combine completely. If there is complete hybridization, then the organisms are identical. If there is partial hybridization, so that some parts of it binds together but not others, then they are related, and then based on that we'll then determine how closely related they are based on the level of binding. But if there is no hybridization, it means they're totally unrelated. So that's one example that is also used, and it's employed in southern blotting and DNA chips and fish. So for the DNA chip, this is how it works. So you would have a DNA chip, as you have in the picture, that will be manufactured mainly by these industries. And the goal is to have single-stranded DNA sequences that, let's say, are very specific to different bacterial species. And so if you have an unknown DNA of a bacteria and you want to find out what kind of bacteria it is, you would then introduce this bacteria onto the chip. You introduce the bacteria onto the chip, and then note that the unknown bacteria that you're looking at has to be cut using restriction enzymes, and then it will be labeled using a fluorescent dye, and then before it's introduced in the chip. But once it's in the chip, then it's then incubated, and then we'll wait to see if there's going to be some hybridization, the concept of complementarity. So for instance, in this example, if there is binding, so if the DNA bind, it will fluoresce, and then the computer will be able to analyze it. And so the red lights are genes that are expressed in normal cells. The green are mutated cells that may be cancerous. And then the yellow are genes that are expressed in both normal and tumor cells. Then there is fluorescent in situ hybridization. So fluorescent in situ hybridization, it uses a dye, a fluorescent labeled probe. If the probe hybridizes with the bacteria of interest, it means that it belongs to that particular strain. And so in this first picture, this is Staphylococcus aureus that is being observed under phase contrast microscopy. And so the DNA probe is then added, and when there is fluorescence, as you've seen here, then it means that we can ID, because we know what the probe is. The sequences are for Staphylococcus aureus strains. And so once it hybridizes, it binds to it because of complementarity, we can then tell that it is that particular strain that we're working with. So this brings us to the end of our lecture on introduction to taxonomy and classification.